Halt exercise disease symptoms with exercise hormone. Halt Parkinson's disease symptoms with exercise hormone. You're listening to Halt Parkinson's disease symptoms with exercise hormone, presented by Hakeem Ali-Bocas Alexander for World Reading Club on the Wisdom app in association with UniquilibriuM. This edition's reading for World Reading Club has a similar title to the title that you see here. The title here on Wisdom is Halt Parkinson's Disease Symptoms with Exercise Hormone. And the article I will be reading from NeuroScienceNews.com is called Exercise Hormone Halts Parkinson's Disease Symptoms. So very similar. I changed up the titles for various reasons. But let's take a look. I'm doing a cold reading, so I had not seen this article before. It just came up in my suggestions through my email subscriptions. And I think that this is very important. It's also relevant to many people. I do know people who have family members who are dealing with Parkinson's disease and Alzheimer's and things like that. So, and also as a part-time fitness trainer, the fact that it starts off with exercise hormone in the original article on Neuroscience News really pops out to me. So I'd like to see what the article has to say here about the exercise hormone. All right, neurosciencenews.com. Exercise hormone halts Parkinson's disease symptoms in the categories of featured, neurology, and neuroscience. Published September 12th, 2022. That's today's date, the date of this recording. Summary. Irisin, a hormone secreted into the blood during high endurance and aerobic exercise, reduces levels of alpha-synuclein and restores movement in mouse models of Parkinson's disease. Source: Johns Hopkins Medicine. Researchers from Johns Hopkins Medicine and the Dana-Farber Cancer Institute in Boston have shown that a hormone secreted into the blood during endurance or aerobic exercise reduces levels of a protein linked to Parkinson's disease and halts movement problems in mice. Parkinson's disease, a neurologic condition that causes people to lose control over their muscles and movements, affects about 1 million people in the US. If confirmed in additional laboratory research and clinical trials, the researchers' study in mice engineered to have Parkinson's disease symptoms could pave the way for a Parkinson's disease therapy based on the hormone irisin. Results of the researchers' tests appeared August 31st in Proceedings of the National Academy of Sciences. Johns Hopkins Medicine's Ted Dawson, MD, PhD, and Dana-Farber's Bruce Spiegelman, PhD, worked together to look into the link between the exercise molecule irisin and Parkinson's disease. For unknown reasons, endurance exercise has long been found to alleviate symptoms of Parkinson's disease. Dawson, whose research focuses on neurodegenerative diseases, including Parkinson's disease, said one of the first clues to the link between exercise, Parkinson's disease, and irisin came from Spiegelman, whose first paper about irisin was published in 2012 in Nature and subsequently in other scientific journals, showing that a protein called an irisin peptide is released into the blood and increases with endurance exercise. In the past decade, other laboratories have found that exercise elevates levels of irisin, and there is interest in looking into the connection between irisin and Alzheimer's disease, as well as Parkinson's disease. To test the effects of irisin on Parkinson's disease, Dawson and Spiegelman's teams began with a research model used by Dawson in which mouse brain cells are engineered to spread small, spindly fibers of alpha-synuclein, a protein that regulates moods and movements related to the brain neurotransmitter dopamine. When alpha-synuclein proteins clump, those clusters kill dopamine-producing brain cells, a key trigger of Parkinson's disease. Fibrous clumps of alpha-synuclein are very similar, says Dawson, to what is found in the brains of people with Parkinson's disease. In the laboratory model, the researchers found that irisin prevented the accumulation of alpha-synuclein clumps and its associated brain cell death. Next, the research teams tested irisin's effects on mice engineered to have Parkinson's-like symptoms. They injected alpha-synuclein into an area of the mouse brain called the striatum, where dopamine-producing neurons extend. 2 weeks later, the researchers injected a viral vector, which increased blood levels of irisin, which can cross the blood-brain barrier into the mice. 6 months later, mice that received irisin had no muscle movement deficits, while those injected with a placebo showed deficits in grip strength and their ability to descend a pole. Additional studies of brain cells among the mice given irisin showed that the exercise hormone lowered levels of Parkinson's disease-related alpha-synuclein between 50% and 80%. The research team demonstrated that irisin also speeds up the transport and degradation of alpha-synuclein via fluid-filled sacs called lysosomes in brain cells. If irisin's utility pans out, we could envision it being developed into a gene or recombinant protein therapy, says Dawson, referring to the widening field of drug development aimed at using cellular genetics to treat disease. Dawson is the Leonard and Madeline Abramson Professor in Neurodegenerative Diseases, Professor of Neurology, and Director of the Johns Hopkins Institute for Cell Engineering. Given that irisin is a naturally produced peptide hormone and seems to have evolved to cross the blood-brain barrier, we think it is worth continuing to evaluate irisin as a potential therapy for Parkinson's and other forms of neurodegeneration, adds Spiegelman. Dawson and Spiegelman have filed for patents on the use of irisin in Parkinson's disease. Spiegelman has created a biotechnology company, Avum Therapeutics, Inc., based in Boston, to develop irisin into treatments for neurodegenerative disease. You're listening to Halt, Parkinson's Disease Symptoms with Exercise Hormone, presented by Hakeem Ali-Bocas Alexander on Wisdom for World Reading Club in association with UniquilibriuM. NeuroScience News continues. Dawson and Spiegelman have filed for patents on the use of irisin in Parkinson's disease. Spiegelman has created a biotechnology company, Avum Therapeutics, Inc., based in Boston, to develop irisin into treatments for neurodegenerative disease. Other scientists who contributed to the research include Tae-In Kam, Hae-Jin Park, Shi-Ching Cho, Yu-Ri Choi, Devanik Biswas, Justin Wang, Yu Shin, Alex Loder, Senthil Kumar, Karap Gounder, and Vanilla Dawson at Johns Hopkins, and Jonathan Van Vranken, Melanie Mittenbuehler, Hyunwoo Kim, Mu A, and Christiane ran at Harvard Medical School. NeuroScience News suggests that you see also, from June 22nd, 2022, uncovering links between determination, grit, and cognitive function under the categories Featured, Neuroscience, and Open Neuroscience Articles. NeuroScience News continues with funding. The research was funded by the JPB Foundation, the Maryland Stem Cell Research Fund, the Mark Foundation for Cancer Research, the Damon Runyon Cancer Research Foundation, and Deutsche Forschungsgemeinschaft. About this Parkinson's disease research news. Author, Vanessa Wasta. Source, Johns Hopkins Medicine. Contact, Vanessa Wasta, Johns Hopkins Medicine. The image in this article on neurosciencenews.com is in the public domain. So that's if you're looking at this research. Original research, open access, amelioration of pathologic alpha-synuclein-induced Parkinson's disease by irisin, by Ted Dawson and others, PNAS. All right. This is a NeuroScience News reading of Exercise Hormone Halts Parkinson's Disease Symptoms is the article I'm reading from NeuroScience News. Here on Wisdom, I called it Halt Parkinson's Disease Symptoms with Exercise Hormone. NeuroScience News continues with an abstract of amelioration of pathologic alpha-synuclein-induced Parkinson's disease by irisin. Physical activity provides clinical benefit in Parkinson's disease, PD. Irisin is an exercise-induced polypeptide secreted by skeletal muscle that crosses the blood-brain barrier and mediates certain effects of exercise. Here, we show that irisin prevents pathologic alpha-synuclein, alpha-syn, induced neurodegeneration in the alpha-syn preformed fibril, PFS, mouse model of sporadic PD. Intravenous delivery of irisin via viral vectors following the stereotaxic intrastriatal injection of alpha-syn PFF caused a reduction in the formation of pathologic alpha-syn and prevented the loss of dopamine neurons and lowering of striatal dopamine. Irisin also substantially reduced the alpha-syn PFF-induced motor deficits as assessed behaviorally by the pole and grip strength test. Recombinant sustained irisin treatment of primary cortical neurons attenuated A-syn-PFF toxicity by reducing the formation of phosphorylated serine 129 of alpha-syn and neuronal cell death. Tandem mass spectrometry and biochemical analysis revealed that irisin reduced pathologic alpha-syn by enhancing an endolysosomal degradation of pathologic alpha-syn. Our findings highlight the potential for therapeutic disease modification of irisin in Parkinson's disease. NeuroscienceNews.com asks you to join our newsletter. Sign up to receive our recent neuroscience headlines and summaries sent to your email once a day, totally free. We hate spam and only use your email to contact you about newsletters. You can cancel your subscription anytime. NeuroScience News posts science research news from labs, universities, hospitals, and news departments around the world. Science articles can cover neuroscience, psychology, AI, robotics, neurology, brain cancer, mental health, machine learning, autism, Parkinson's, Alzheimer's, brain research, depression, and other topics related to cognitive sciences. You've been listening to NeuroScience News Reading, presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM.